Good morning, everyone. I'm Simran Da Costa, and on behalf of Altum Technologies, I'd like to welcome you all for one more very interesting virtual session. So, our today's session is entitled as In Vitro and In Silico Screening for Antiviral Natural Compounds. So, before we begin, a little bit about natural compounds and the use of bioinformatics. Now, we see that natural compounds exist in plants or other biological sources, and they've been utilized as therapeutic products since prehistoric times. And the state of the art technologies in biological research allows us to examine different proteins and biomolecules that interact at the molecular and cellular level. So, the application of these technologies in our search for therapeutic products, especially for the vast trove of natural sources, is quite promising. And these efforts can further be optimized and made efficient by the use of in silico tools. So, the developments in the domain of bioinformatics have brought out many tools to optimize the process of identification of therapeutic molecules. And the synergistic use of in vitro and in silico tools and techniques is the way to go forward to identify useful and therapeutic compounds that are hidden in natural resources. So let's understand the perspectives of the use of bioinformatic tools and for identifying natural antiviral compounds from our speaker of the day. So it gives me immense pleasure to introduce Dr. Anbu Kumar Karupanan. Dr. Anbu Kumar has obtained his degree in veterinary medicine from Madras Veterinary College, Tamil Nadu in 2000. He has been working in the areas of microbiology, immunology, and vaccinology since 2000. He has obtained his master's degree from the University of Kentucky, Lexington, US in 2003, and PhD from the National University of Singapore in 2012, where he has worked on viruses and vaccines. He has also worked on immunology and infectious diseases at the University of Alberta, Canada, and College of Veterinary Medicine, USA, as a postdoctoral research fellow. Presently, he's working as an assistant professor attached with veterinary microbiology and bioinformatics department at Madras Veterinary College, Chennai. His interests are in the areas of infectious diseases, vaccines, and bioinformatics, and he has published more than 30 research articles indexed in PubMed and holds one US patent on surface display of antigenic proteins. Now, without taking much time, I'd like to hand over the platform to our expert. So, welcome, sir, and I invite you to take over the session.
Thank you. Thank you for, first of all, I would like to thank the people at Altum for providing me this opportunity to speak in this very good forum. I think this is a very good effort to seek natural compounds as lead molecules for designing therapeutics and drugs. And also, I'm happy to say that we are at our facility. We have the BioVias Discovery Studio, which was supplied to us through Altum, and we are making very good immense use of it. So, coming to the topic of today's lecture discussion. So I'm, as Simran has indicated, I'm basically trained as a microbiologist, and all my work has been in the area of microbiology and virology, a little bit with vaccinology. So, the talk that I'm going to give you today is more about the identification of the lead compounds for antiviral property. And I'm going to elaborate in the first part about work that I did 10 years ago in 2012 in the National University of Singapore. And I'm also going to compare that with what we have done recently and show you the parallels between these two. So let's get into the topic. So recently I came across this article in the BBC about maybe the 1 month ago. The date is there, 2nd of May. And the article was discussing about a group of scientists who are observing orangutans in their natural settings in Indonesia, And there they observed that one male orangutan, uh, which fought with other males or other members of his troop, sustained an injury on the below his right eye. As you can see that in this figure, partially hidden by that play arrow, but still you can see that there. And this guy went on to chew some of the leaves from the environment. It's actually called the akar kuning, that plant. And he chewed the akar kuning plant and took the juice from the plant and also that chewed material and applied it on his wound. And he repeatedly did this, and within 1 month, the wound, which looks really horrible, the wound got healed completely, and he was back to normal. So this shows that there is an instinct among all mammals to look for such natural remedies existing in the environment to heal themselves. And as a veterinarian, I've noticed this in cats and dogs also. In raised in our country or in rural places, I've seen this. They go and eat certain leaves and certain grass blades to once in a while. They are actually not vegetarian by habit, by their diet. They are omnivorous animals, and mostly they like because of their canine origin, they like meat type food. But still, they go and seek out certain plants to heal themselves. So what I'm trying to tell here is this instinct for looking. This is actually an instinct. I would say it an instinct. Instinct to look for remedies in our environment is very primitive, and humans have been doing this use of medicinal plants for time immemorial. Since prehistoric times, we have been using that. So it is no wonder that now there is a renewed interest in looking for remedies and therapeutic molecules in natural compounds. And if we look back in history, a lot of drugs that we are currently using, they are originating from plant or natural sources. For example, aspirin, one of the foremost drugs used for anti-inflammatory and pain remedy. You can the aspirin was obtained from willow tree originally, and then now we synthetically do it. And same with morphine. Morphine is a painkiller. Before the advent of morphine, surgeries were really painful, but morphine and other opiate drugs, it belongs to opiate group of drugs. Morphine and other opiate drugs are now commonly used, and they are originating from a plant, the poppy plant, or scientific name is Papaver somniferus. Similarly, so many other drugs like your antibiotic penicillin is from a fungi. We all know the story of how Alexander Fleming serendipitously discovered this drug. And colchicine, which is a chemotherapeutic agent, atorvastatin, which a lot of people take now to control their cholesterol levels, or other analogs of the statin, and artemisinin, the famous malaria drug. And this discovery of artemisinin by a scientist, Chinese woman scientist, Dr. Yu of fetched her a Nobel Prize, as we all know. And so the list goes on and on and on. So if you look in the archived popular scientific field, you can see that there is a surge of, you can see on the left side bottom panel, you can see that from 1958 to 2024, there is a big surge in the publications which talk about the natural compounds. Especially, I did this search yesterday when I was making the PowerPoint, and used the search terms antiviral, natural, and compound, and it fetched me this profile. So you can see that there is a huge explosion of publications in the natural compounds, in looking for therapeutic molecules in natural compounds or of other natural material. So back in 2012, I was when I was in the National University of Singapore, I had the chance to interact with one of the researchers who was working on this enterovirus 71 and also dengue virus. He had some leftover material from a natural compound library. It's a physical library. So there is a company which sells library of compounds in the physical format, in liquid format. And they had about roughly 500 compounds, and he tried some screening for dengue virus, and he had some leftover material from that. So I was interested in looking at for an inhibitor for this porcine reproductive and respiratory syndrome virus. This is a very common virus in pigs, and it causes a lot of devastation in pig farming. And so this virus, though there are vaccines, the vaccines are not completely effective, and addition of antiviral compound in addition to the vaccines would be really helpful in managing this viral infectious disease. So I obtained this library from him. So this was from a company called Enzo, and I checked this yesterday again, and this company is still selling this library. So it is a library called as Screenwell Natural Product Library, and it has roughly 520 compounds, and this 520 compounds are composing of terpenoids, uh, peptide, so flavones, alkaloids, macrolides, isoflavones, and whatnot. So many different natural compounds are there, and all of them are in liquid format, dissolved in dimethyl sulfoxide at a concentration of 2 milligram per ml. And this is supplied in a 384 well format. So they give many plates, and 2 milligrams of that is 1 ml of each liquid is given. So so 500 microliters per well it is given. So whatever kind of assay you are going to develop, you can directly pipette the compounds from the supplied material to your assay format. If it is designed in a 384 well format, it's going to be very easy, and it can be done automatically using robotic arrangements. So the idea for us was to identify natural compounds that can inhibit the porcine respiratory syndrome virus. So the assay was set up. So the idea was to screen for inhibitors that can either prevent the virus from entering the host cell. So we got a cell line which allowed the replication of the virus. It is actually of a respiratory origin. It's a macrophage alveolar. It's the alveolar macrophage, that is macrophages which circulate inside the lungs. So these alveolar macrophage derived cell line called MARC 145 cells were used for this assay. And basically the assay was set up to screen whether the virus, to prevent the identification of compounds which prevented the entry of the virus into the cells, or after the entry of the virus into the cells, any of the compounds, could they inhibit the replication of the virus. So we were looking at two things. And to look at the former, to look at the inhibitors for viral entry, we seeded the cells, then added the compounds next, waited for some time. Then once the compound had permeated into the cells, then we infected them with virus too, and then checked for the infection signs of infection. In the next objective, we were infecting the cells with the virus and then adding the inhibitor 12 hours after infection, and then we checked for the virus load after another 48 hours. So either way, we are looking for either the same assay was based on the sequence of addition of the virus and the inhibitors. We were looking for either the inhibitors of virus entry or the inhibitors of viral replication. And once the virus was started replicating in the cells, they were the replication of the virus was identified using a immunofluorescence assay, and it was also done in a automated format. So this is the automated machine. So it is called Cellomics scanner. Now it is under the Thermo Fisher management, and this the Cellomics scanner can screen 384 wells or 96 up from 96 wells to up to 384 wells in many different formats. It can see, and it can see many wavelengths. So we were using a secondary antibody to look at the monoclonal antibody to detect the viral proteins, and a secondary antibody to detect the monoclonal antibody, which was in the red wavelength. And so this software that was attached with this machine was capable of returning the level of virus infection in each cell by looking at the viral proteins. And also we used DAPI to look at the cell nucleus. So we know whether the cell nucleus was there, whether the cells were healthy and there, and also if the virus was there. So you can see in this screen that the topmost panel, two panels, they show the mark infected cell, that is cells without any virus infection. You can see that the DAPI signal is clearly seen, but the virus signal is not seen there. And in the next panel, we infected the cells. On the left side, you can see the DAPI indicating of the different nuclei in the cells, and also on the right side, you can see that the virus infection gives rise to a signal in the red wavelength. And in the lowermost panel, you can see that though the cells are infected, they were added with a chemical inhibitor called mycophenolic acid, which is a nucleoside which can inhibit the synthesis of RNA basically. So this being an RNA virus, the virus could not synthesize its further synthesize its genome after infection. So there was very poor viral replication. So you can see that the assay is working well. So basically, we had an assay which can tell us the level of replication of the virus in the Mark 145 cells, and we were planning to use this with the different set of different inhibitors before the infection or after the infection to look at the prevention of viral entry or inhibition of viral replication respectively. So this assay was done in a 384 well format, so it's a high throughput assay. And when we did our experiments, we were able to identify at least 19 compounds which were inhibiting the virus replication. But none of them were able to prevent the entry of the virus. Unfortunately, none of them were able to prevent the entry of the virus. That was kind of expected, because only antibodies can do that. Antibodies bind with the virus and neutralize the virus, thereby preventing the entry of the virus into the cell. But in our screening, we could not identify any of them which blocked the entry of the virus. We were hoping that some of the chemicals could inhibit the the cytoskeletal mechanism associated with the endocytosis or receptor mediated entry of the virus, stuff like that. But none of them worked that way. But 19 compounds they inhibited the virus replication after the virus infection was set in. So they were able to suppress the virus infection. And among these 19 compounds, some of them were cytotoxic. So cytotoxic in the sense, even though they inhibited the virus replication, the count of the infected cells also went down. So they were having both activities. So perhaps they were doing the inhibition of the virus replication by suppressing the vitality of the cell. There were 4 compounds which we finally narrowed down to, and these 4 compounds were able to inhibit the virus replication at sub-micromolar levels. So these were bufalin, ouabain, forbolester, and valinomycin. So these 4 compounds which were supplied in the library, they were able to inhibit the virus replication at sub-micromolar concentrations. And here you can see the panel of our bufalin. So the bufalin, which is an steroid molecule, it was able to completely inhibit the virus replication, as you can see, at 8 micromolar and 1 micromolar levels. At 0.1 micromolar level, it was inhibiting to a good extent. I think it was around 4 to 7%. I don't remember well. We will see that in a minute. So it was inhibiting for less than 1 micromolar. And same case with valinomycin. This valinomycin is a cyclic peptide. So this cyclic peptide had a very wonderful profile of inhibition. You can see even at 0.1 micromolar, it completely inhibits the virus replication in the cell culture model. And next, this forbolester, 12-deoxy-4-ball phenyl acetate, 20 acetate. This one, it was able to inhibit to a great extent at 1 micromolar, but at 0.1 micromolar, there was not complete inhibition. So these are the 19 compounds that we were able to identify. So here I'm showing you the 10 compounds at different levels, and you can see on the left hand side, you can see the level of the surviving cells, that is with controls, and the different compounds added. And on the right side, you can see the virus signal. So you can see that the number of cells were not compared to the treated cells that were not treated with any compounds, but just with 0.1 molar, 0.1% DMSO. The signals were compared, and you can see that bufalin, it reduced the concentration of the cells, as well as the concentration of the virus, and at the same time it did not reduce the concentration of the cells. So the viral signal was brought down to 17% of the untreated cells, whereas the viability of the cells was at 96%. I request you to focus at the second row. So you can see at 10 micromolar, the viral replication level was 0, and at 1 micromolar, it was 2, and at 0.1 micromolar, it was 17. Similarly, in the viability of the virus infected cells was 78% at 10 micromolar of the compound, 94% at 1 micromolar, and 96% at 0.1 micromolar of the compound. So it was having a good safety profile at the cellular level, as well as it had a good inhibition potential for the replication of the virus. So in our studies, we were able to identify 4 such compounds. So the first one is bufalin, which is a steroid molecule, and a furbal ester, and ovabain, and valinomycin. Valinomycin is an ionophore. So all these 4 compounds, they had a good inhibition at 0.1 molar concentration. You can see the bufalin was at 17% of the untreated cells. Furbal was at 36%, ovabain at 2%, and valinomycin at 4%. So at 1%, sorry, 1%. So at the same time, the viability at 0.1 micromolar of the compounds were more than 95% in all the cases. So next we went on to estimate the inhibitory concentration 50. So IC50 dose response curve was set up, and all of them had a sub micromolar concentration of the IC50 with good standard deviation. There was not much dispersion. And the identified 4 compounds were further evaluated to check whether the compounds were capable of inhibiting the virus replication at the early stage or at the late stage. Because in clinical settings, most of the times, whenever we want to go to treatment with antiviral compounds, the patient, or in our case, the animals already have the signs of infection. So it would be very useful to have a compound which can inhibit the virus even at the later stages of virus replication. So we infected the cells and added the inhibitors at different times after the inhibition of the onset of inhibition. Onset of infection. So you can see the three columns. So the one in the column 1, it is 0, which means a compound was added along with the virus. In the column 2, you can see the inhibitors were added 2 hours after the infection of the cells, and the column 3, the inhibitors were added 8 hours after the infection of the cells. So you can see that bufalin, which is in the row 1, and ovabain, valinomycin, and our control mycophenolic acid, they were able to inhibit the virus replication even at 8 hours post-infection. But your furbal esters did not do that. They were able to inhibit only in the early periods of the virus infection. That is, at the time, if you add the furbal ester at the time of infection, it is inhibiting the replication, and 2 hours after the infection, it's still inhibiting the virus replication to 2% level. But if you add the inhibitor at 8 hours after infection, the inhibition is only up to 68% of the untreated cells. So it is not working in the late period of infection. So with these assays, we were able to identify compounds which were able to identify all the virus replication to a great extent, even at say 8 hours post infection. We didn't go beyond that at that time because of the lack of availability of the compounds. Because I was using a leftover library from another scientist, so this is all we had for that at that moment. However, it was a very promising results. These results were very promising. But the caveats that we took away from this study are, all this study was done in a cell culture model. The Same thing may not apply in an animal model, because the animal has a lot of multiple different cell types. In addition to that, it has a circulation, it has hepatic portal circulation, it is having the metabolic organ, the metabolism will be there, liver will degrade it, kidney will excrete it, all those things are there. So these were very promising, these four compounds, at least the three compounds, bufalin, ovabane and valinomycin, they were very promising compounds, which were capable of inhibiting the PRRSV in a biological model, but although at a cell culture, cellular level. So the next we wanted to take it to animal level, but at that time I got a job here in India and I moved on to this assistant professor post. So we were not able to complete the animal model trial. But the thing is, the next level of things that we need to look at from this study are what will happen to the compound inside the animal. That is the ADME studies: absorption, distribution, metabolism, excretion. All these studies. And what about their bioavailability? Are these compounds able to cross into the different organs, and because this virus obviously affects the respiratory system and the uterus, the reproductive system. So will these compounds be able to reach into these organs, and will it be bioavailable in the cells in these organs? And what about their toxic profile? What about how will the compound affect the other cells and other systems, organ systems and processes, biological processes in the cells, or the functioning of the different organ systems? How will they affect these things? So all of these things have to be further followed up. But nowadays, with the availability of all the bioinformatic tools, all this ADMA, absorption, distribution, metabolism, excretion, including toxicology, everything can be done or predicted using bioinformatic tools. So at this point, I will also like to make a note that, so this was a assay that we set up for looking for viral inhibitors, natural compound inhibitors of viral replication, a particular virus, using a cell culture model. But if you are a scientist who is working on, say, let us say diabetes or any other metabolic disease, or maybe some other kind of cancer or some other thing, you can still set up your own assay. The setting up, to me, from my perspective, I think the setting up of the assay is the most important thing. So whenever you set a proper assay with all the controls, like in this case, we had designed the assay to look for inhibitors which can prevent virus entry and inhibitors which can prevent virus replication. And we were fortunate enough to have all the necessary high-end equipment, because the National University of Singapore is a well-equipped place. They had the Celomics scanner, which is a very expensive equipment, and we were able to afford to buy all the monoclonal antibodies and all the reagents required for setting up this high throughput assays in a 384 well format. So similarly, whenever you plan to design such high throughput experiments to identify natural product that suit your purpose, or that you are looking for, you need to set up a proper assay. So this designing of the assay is the most critical thing. And once the assay is set up with all the proper controls and all the necessary rigor, you are going to definitely hit some good leads for whatever the study that you are conducting, maybe metabolic disease, or maybe cancer, or even geriatric prevention of aging, whatever. You can set a assay that you want. And this particular assay we set up was at cellular level. You can also set up an assay at molecular level with in vitro molecular assays with purified proteins and buffers and stuff like that. You don't have to go with a cellular assay. You can also go with further go down even more specific with a molecular assay. Even in this case, for example, since we were working with this is a RNA virus, we could have looked at a in vitro assay in which the RNA polymerase of the virus was, the inhibition of the inhibitory potential of the different compounds in the library on the RNA polymerase of this particular virus could have been devised. But that would require a bit more, elaborate setup, understanding of the capacity to isolate and produce the RNA polymerase of this virus in an in vitro system and to make it work in an in vitro system, then you have a very beautiful assay which can directly target the inhibitors which are going to bind and interact with the virus RNA polymerase. So again, I'm just saying that setting up of the assay is the most crucial part. So next, I was talking about the bioinformatic tools that could be used for further characterization of the identification of the identified lead molecule. So in our case, we identified bufalin, ovabine, valinomycin, the three new compounds, and forbolester, not very good at later stages of inhibition. So this valinomycin is a cyclodepsipeptide, cyclic peptide, and it's already known antibiotic, which is isolated from streptomyces strains, and it is it also acts as a inophore. So all this information was available in the PubChem, the website PubChem. I think which all of you with interest in this field probably know. And I was also able to isolate the chemical canonical smiles for the compound. So with the availability of these smiles structure, we can go on and do further analysis about the ADME and that is the absorption, the pharmacokinetics and toxicology profile basically. So this was the canonical smiles for the next compound, bufalin. So interestingly, bufalin is already known to the Chinese medicine. In Chinese medicine, they call it shansu. So it's of steroid origin. It's a steroidal molecule, and it is found naturally in the venom from the Chinese toad. So some frogs, they secrete this venom on their skin. So this Chinese toad called Buffa gargarizans, it secretes this bufalin molecule in their skin, and the Chinese people, they have used this as a traditional medicine for a long time. So probably just like artemisinin, this could also serve as a potential inhibitor for this virus. So once the smile structure is obtained, you can take the smile structure and you can plug it into different tools to identify the different properties, the pharmacokinetic properties and toxicology properties of the molecule. So this PASS online server is a very interesting tool. It can predict the biological activity of different molecules. So if you have the smile structure, you can plug it in and it will show you the probability of activity which is shown in the green, and also the probability of inactivity of a particular compound against a particular condition. So you can see that this bufalin is having a property as a respiratory analeptic. And similarly, if you go to the lower end of the box, you can see that it is having it is predicted to have antiviral activity because it's shown to be having some activity against the influenza virus, and the probability of activity is given as 0.694, and the probability of inactivity is 0.006. So it is having a good probability that it may act as a antiviral agent. So there is another website called Swiss ADME. So all these websites are freely available, and the only problem is you have to go to different websites and plug in your smile structure and get all the results. So this Swiss ADME is another website which can give you the pharmacokinetic properties like the physicochemical property, lipophilicity. All these different biochemical properties are available there, and it also gives them as a radar plot for understanding the pharmacokinetic properties of the drug in a much better way, the lead compound that you identified in a much better way. And also it can give you a boiled egg profile which shows you to which different bodily compartments the drug can penetrate. So this bufalin, obviously it seems that it can penetrate the blood-brain barrier. So typically in any mammalian organism and higher vertebrates, there are two barriers which are hard to breach by the drugs. One is the blood-brain barrier, because it is a very precious organ, nature has evolved to bring about a lot of barriers for the entry of biomolecules into this blood-brain barrier, which is surrounding obviously the brain. So this drug has the capacity to go into the blood-brain barrier. The other barrier is the placental barrier. So placental barrier is the ability of a drug to cross into the placenta, and placenta, that is the placenta is the organ which is formed during the pregnancy. So if the placental barrier is breached, if the drug is able to cross the placental barrier, it may reach the fetus, and this may sometimes have inadvertent effects on the developing fetus. So you can predict all these the things using this tool, the Swiss ADME. And again, there is another tool called Admet Lab. So the current version of the Admet Lab 3, it also has all the, if you plug in your SMILE structure, so once you identify a lead compound and take the SMILE structure and input into this website, it can predict a lot of other this kind of properties like the pharmacokinetic absorption, toxicity profiles it can predict. And it can give you in terms of a visual green, amber or red signal, just to make the profiling easier. And there is another website called for prediction of toxicity, this Protox 3 website. It can predict the systemic as well as cellular toxicity of the cellular level toxicity of the compounds that you are plugging in. For example, in this screen you are seeing that the bufalin has a high risk for respiratory toxicity, immunotoxicity, and cytotoxicity. But fortunately, in our cellular assay, it did not have much toxicity to the MARC145 cells, which are derived from pig lung tissue. That is, it's a alveolar macrophage derived cell line. So, however, here the server, the Protox server indicates that it could potentially have a cytotoxicity. But if you look on the rightmost panel, it says the probability is only 0.82. So probably it's not really a risk in pig cells. Okay, so there is always a little bit of mismatch between what we see in the prediction servers and what really happens inside the the living cells or in the living animal. But however, it's a most of the times these predictions have good indication. So the BioVia Discovery Studio software, which we have in our library, in our laboratory, it has all these things: the absorption, the ADMET properties, that is the absorption, distribution, metabolism, excretion, and toxicology profiles. Everything is built into this software, and it can do the predictive toxicology, and in addition, it can also do the quantitative structure activity relationship based on the structure of the compound that you are working with. So if you go to PubChem, it will give you the structure of a lot of different compounds, and so based on that you can do a quantitative structure activity relationship assessment. So it has been a very useful tool in our laboratory, and we use it to train a lot of students who come here for training. Postgraduate students, even sometimes undergraduate students come and get trained, those with interest in the area of drug discovery. So next I'm going to move on and show another piece of work which was done recently, last year, in our laboratory by a student for a project work. So this study was focused on another veterinary virus called Newcastle disease virus. It is a major pathogen for birds, especially in farmed chicken. It is in farmed and domestic chicken. It is probably, you can say, the number one disease in chicken. Again, this is a RNA virus with very few proteins. You can see some 7 or 8 proteins are there in this virus, and the most important, the only protein in this virus which is responsible for the virus replication is the polymerase. It's a RNA dependent RNA polymerase. So it uses the viral RNA genome as template and produces many more copies during this infection phase. And so we were planning to identify any inhibitors that can bind to the any potential inhibitors that can bind to the polymerase. So any compound that binds to the polymerase may have a inhibitory activity, or may sometimes even have a potentiating activity. But we don't know that. But we are looking for binding partners, good binding partners with this polymerase protein. So the previous study, at the end I told you that the study which was conducted at a cellular level could be further refined by doing a molecular assay based study. So here, instead of here, we were using that principle and looking at binding partners, natural compound binding partners for the viral RNA polymerase using in silico analysis. Okay, so this RNA polymerase was already the structure for the RNA polymerase of this particular virus was determined using cryo-EM. So this cryo-EM determined three-dimensional structure at 3.4 Armstrong, I believe, 3.5 Armstrong resolution is available in the PDB. Again, this protein database is a very wonderful resource where you can go and search and find the molecular structure, 3D molecular structure, determined structure, either by cryo-EM or NMR or by other circular dichroism or whatever physical study. A lot of determined structure are available in this PDB database. So fortunately for us, our this Newcastle disease virus polymerase protein structure was available in the database at 3.4 Armstrong resolution. So we went there and downloaded this structure, and so along with this structure, we went to this database maintained by the Ayush department called IMPACT, the Government of India Ayush department. So this IMPACT database again is a wonderful resource. It has the 2D and 3D chemical structures of all phytochemicals in different Indian plants. So in India we have different traditional medicine systems, starting from Ayurveda, Unani, Siddha, and whatnot. So many different traditional medicinal practices have been ongoing in this country for thousands of years. So all this knowledge has been brought to into this website. So this website lists the phytochemicals in different plants. So you can browse into, if you go into this website, you can browse the phytochemicals by looking at the different plants. You can either put in the plant name, and even within the plants you can choose that a particular part that you want to look at, either the bark, the root, the leaf, whatever, the flowers, whatever you want, you can look. And it's also classified under the mode of act, the target action, that is like a cardiac glycoside or respiratory bronchodilator, something like that. You can put, and then you can retrieve medicinal plants which are having that kind of activity. So this is a very wonderful website. So in that website, we chose the neem plant. So the biological name Azadirachta indica, the neem plant. So there were around some 2,000 or 3,000 phytochemicals listed from this neem plant, and they were all listed under different categories: bark, the neem fruit, the neem seed, the neem leaf or neem root, etc., etc. So many different categories were given. So I'm showing you the molecule called nimbayal. So the nimbayal is found in the bark of the neem plant. You can see it there. And so here this nimbayal is a steroidal molecule, and it has the smile structure and other chemical structures. The 3D and 2D structure are also given in this particular website. So we went ahead and we collected a list of different natural compounds from this website, the chemical structures and the 3D structures of the compounds from this website, and used the BioVia Discovery Studio software, especially the libdoc module. So the BioVia Discovery Studio, it allows you to do a in silico docking analysis. That is, you can use the protein structure along with the biomolecular ligand structure to see if there are any interacting pockets, any pockets on the target molecule to which the ligands that you input can bind. So this way we screened so many different compounds from the neem plant, which were screened against this polymerase protein structure, which we downloaded from the PDB. So in our study, we identified some 20 or 30 molecules. So, but this libdoc module, it gives you the interaction score along with the free energy of binding. So this interaction score was ranked, and we selected a few compounds which were having good interaction score as well as a low toxicity profile using. So all of this was done in the same software. So we selected some 4 molecules, and you can see the software even returns the, because you can see the structure of the syringic acid which is given here on the right side panel. So this interaction of this syringic acid molecule with different amino acids in the three-dimensional structure of the viral polymerase, that is the polymerase of the Newcastle disease virus, is given here. You can see the different molecules in the interaction of the different molecules in the syringic acid with the different amino acids in the polymerase protein are elaborately given in the software. And the optimization, the binding study can be further refined by optimization of the the optimization of the binding examination, and we can further fine tune this. So in the same manner, we were able to identify some 5 or 6 compounds which were really having a good binding profile and also low toxicity scores and good bioavailability. So that is also important. The in silico identified molecule should have a good bioavailability. That is, drug likeness properties were examined. Without this compound being able to reach the reach the animal's target tissue, it's not going to be of much use in further assays. So all these properties were examined, and we have arrived at a set of compounds which are able to bind to the viral RNA polymerase to a great level. But whether these binding partners have an inhibitory property or any other property, these things can be verified only using a wet laboratory confirmed only using a wet laboratory assay, using a biological assay. But at the moment, we have arrived at some compounds which are really promising. So we are so now we this kind of study we quite often conduct in our laboratories is using different viruses, and but all these the results of all these studies are very promising, and we are actually looking for further funding to expand these studies to see if the results which have turned out using our in silico predictions have any biological relevance. And the thing is, wet laboratory approach alone to wet laboratory assays to screen for natural compounds is going to be very expensive, prohibitively expensive, and cannot be done for every disease model or every infection model. But the in silico approaches are highly synergistic with the in vivo or in vitro approaches. So the in vitro approaches, lead compounds identified in vitro approaches can be further assessed for their toxicity and other profiles, and also at the same time you can also use the in silico prediction to narrow down on the molecules that you would want to explore using in vitro assays or cellular molecular assays. So this way the utility of the in silico and in vitro screening are synergistic. Okay, thank you for the opportunity, and I will be happy to interact with the participants.
So thank you so much, sir. This was a very knowledgeable session. So it's time to take some questions now. So the first question is how to perform virtual screening.
Okay, the virtual screening can be done using a lot of proprietary softwares and a lot of free software available on the internet. So the common proprietary softwares are the Discovery Studio by the BioVia, or there are other proprietary software also like Schrodinger and others. I'm not very familiar with many of these. I'm basically a microbiologist by training, so I'm a little bit not very familiar with the bioinformatics side of these things. Our students helped us. And if you want to go to the internet, there are a lot of free sources also. I think Autodoc Vina is one popular software which is freely available. But if you really want to work with Autodoc Vina, you have to be very conversant in Python. So it requires a lot of coding skills, and it's a little bit more difficult to work with compared to the Discovery Studio, where you have everything in a graphic user interface, and all the different modules are available in one software package. So it depends. So the virtual screening, basically for the virtual screening, you need the structure of the molecular structure of the target protein that you need to work on, and also the molecular structure of the ligands that you are going to pair with this target protein. So the molecule, the protein database, PDB database, will give you the molecular structure of the predicted, the determined molecular structure of the different target proteins. And you can also run a blast P, the common NCBI blast. You can paste your protein sequence in the blast P portal, and then you can search against the PDB portal. That option is available. So input your sequence in the blast P and search against the PDB database. That can be done to identify the structure of your protein. If the structure of your protein is not already available, the determined structure, you can do a homology based modeling. That homology based modeling or de novo modeling can be done with a lot of software, including your Discovery Studio. So then you can go to the PubChem to search for ligands which you can dock with the protein structure. Okay, so all these are available in internet. All the protein structure and the ligand structures can be retrieved from the internet databases, and the docking can be done with proprietary or free software.
Okay.
Okay, does it answer your question?
I think it pretty much did, sir.
Okay.
So the
How
next
to?
question is
Mhm.
how to assess the effect of Ayurvedic formulations which contain multiple active compounds.
Okay, so if you want to assess multiple compounds, you will have to only go for a cell-based assay or a animal-based assay, an animal model or a cell culture model. If you are going for docking, that is the in silico interaction analysis, then you have to choose a particular target molecule and a particular ligand molecule, because as you said, the formulation will contain multiple active compounds. So you have to pick each active compound and look for the target which the active compound may bind to. So for that, you can go to the PAS server, because the PAS online server, it has the capacity to predict the biological activity of each compound. So once you try and see what kind of activity this active compound has, then you can probably predict or guess the target. You can make an educated guess on what protein this particular compound can bind, and then you can try to see the interaction. But again, uh, without the specific knowledge on the target protein which you are looking at, or the idea of what protein you are looking for, it's very difficult to assess the a particular interaction between the active compound and the target protein. So basically, you need to know the target protein.
Our next question is which target proteins can be used for anti-cancer activity?
Oh, cancer is not caused by a single mutation. It starts with a single mutation usually, but there are multiple proteins usually involved. Proteins involved with the cell cycle checkpoint, proteins involved with the cell cycling, DNA repair. So many proteins are involved. It starts with one, and as it progresses, many mutations are accumulated. So there is no one particular protein, but there are certain types of cancer which are implicated on certain genes. But even there, I think it starts. For example, breast cancer is associated with the BRCA gene. Prostate cancer is associated with the PAR4 gene. It starts with one particular gene, then it progresses on. So I think when people look at cancer, chemotherapy for cancer, they do a lot of assays, molecular level assays, and look for the aberrations in a lot of cell cycling, cell signaling pathways. What kinases are activated, what kinases are not activated, what proteins are upregulated, what proteins are downregulated, and stuff like that. So uh, there's no it's not possible to put a finger on a particular protein and say that this particular protein is going to cure the cancer. In targeting this particular protein is going to cure the cancer. It's not like that.
Our next question is, how can we check the novelty of compounds?
Oh, by novelty, I assume you mean the patent rights and the intellectual property rights, right? So you can always go to the PubChem and plug in the chemical structure of your compound and see if I think the PubChem, it has a link for intellectual property associated with a particular chemical structure. So you can always go to the PubChem and do the search, and then you can see whether the compound is already patent covered by property rights, intellectual property rights.
Our next question is, does Discovery Studio help to predict the admit property?
Yes, of course. I think there is a particular module called TopCat that is very helpful in this. And I think, yeah, the version that we have in our laboratory is the version 2020, BioVia Discovery Studio version 2020, and we are able to do all the the admit prediction in our software.
How to identify in general which compound poses antiviral activity?
Well, like I said, you can go to the PAS server or any other, a lot of bioinformatic databases and online tools are there. Even proprietary tools are there. So they can predict based on the chemical structural similarity, they can predict the function of a particular compound. Even PubChem, to start with, the PubChem can do that. Uh, but again, you have to verify that in a biological assay to make sure that it is having this property.
The next question is, will bufferine is not extracted from plant? Is there any alternative source to get a similar compound?
Yes, if you look at the story of aspirin, which was originally extracted from willow tree, now it's made synthetically. And similarly, artemisinin, the drug used to treat malaria, is now synthetically made. And so on. Like so, similarly, even there are semi-synthetic penicillins. So all these compounds are lead compounds. They're called lead compounds. Once you identify a lead compound, then people do something called as pharmacophore modeling. Pharmacophore modeling is looking at the biological molecules or the ligands which have similar activity against a similar same target, similar activity against the same target. So based on this, they come with a common set of, how do you say this, common set of steric and electronic, I think that's what they use, steric and electronic features which are found in all these ligands, and that is called a pharmacophore. That is the core of the all the ligands which are able to do the same activity in the same protein. Then this pharmacophore can be used to do chemical synthesis to make a synthetic drug. It is possible.
The next question is there any simple test to check the antiviral activity of a medicinal plant leaf?
Okay, if you want to look at the activity of a medicinal plant leaf, go to the impact server. The impact server, you can select the biological name of the botanical name of the plant, then look at what compound you are looking for, and then you can get that particular compound. The smile structure will be readily available there. The smile structure you can put in the PubChem or PAS online server, and then you can see what kind of activity it predicts. So all these things are again predicted with certain level of probability, and all these predictions are based on the already available data. So there are a lot of compounds already in the literature and in the domains for public databases and private databases. So these chemical structures are associated with certain biological activity. So based on your chemical structure that you are putting in, the other activities will be retrieved by these servers using different algorithms. So it's all a prediction based on the chemical structure of your compound.
Okay. I think we are done with our questions, sir.
Okay. Thank you all for your kind attention,
Thank you, sir.
and I hope all of you are able to delve into this world of natural compounds, because we have this wonderful impact server. I highly recommend you all of you to go and have a look at this impact server, because there are so many different chemical structures for so many different medicinal plants available there. Then also go to the protein database, the protein database PDB. So there the structures of so many different proteins are available there, so you can try to play around with these things. Thank you all. Thank you, Dr. Praveen.
Thank you, sir. So it's now time to felicitate our speaker of the day with a memento and a certificate of appreciation. So thank you so much once again, sir, for giving your
Thank
time
you very
and
much.
for the great interaction. It was really a pleasure to have you with us here today. And
Same here.
also, I'd like to, yeah, and also I'd like to thank all the participants for being a part of this session. So, yeah, thank you so much, sir.
Thank you all.
And guys, please stay tuned with us. Please leave a like. And to get updates for more informative sessions like this, please subscribe to our channel and follow us for more upcoming events. So, thank you so much, everyone. Thank you so much, sir.
Thank you. So I can log off now.
Yes, sir.